Hello, everybody. This is going to be a voiceover presentation of my lecture on transcription and translation, otherwise known as protein synthesis. You should be watching this video, this Zoom video, on the heels of the two that I produced previous to this, and that's on cells and molecules, part 1 and part 2. All right, so let me start my screen share of the PowerPoint. So in this particular lecture, we're going to talk a little bit about how DNA goes about getting its job done. In other words, how it produces information and delivers that information to its manufacturing facilities, and then how those manufacturing facilities turn that message into the actual products, which are proteins. First, we need to sort of get orientated to where all this is happening, the cellular geography, all the transcription that's happening. In other words, the copying of DNA into RNA, our message, is happening within the nucleus of the cell. Right? So DNA is copied into RNA. Then RNA is delivered to the ribosomes. And the ribosomes are actually working on a structure called the endoplasmic reticulum. And then the ribosomes read the RNA message, and they sequence amino acids together, and a growing chain of amino acids becomes a functional protein, either the structural stuff for your body, or it becomes the enzymes that catalyze important reactions. They're really the tool builders. They're the maintenance items of the cell. Okay, so we'll go through this in a more detailed way, and to sort of talk about some of the nomenclature that's really important, that's sort of attached to some of these elements in the picture that you're looking at right here. So first of all, like I was talking about, really the end product of this is what's important. And of course, the end product of the structural stuff. So I want you to think about things in your body. I've mentioned things like hair, and skin, the collagen in the skin, but also bone is a lot of proteins. Muscle, majority all your muscle, is myosin and actin, and all sorts of important protein filaments. And also the hormones of the signaling, the items in which communicate between cells and major systemic processes are produced by proteins in which DNA codes for. And of course, again, the all-important enzymes, those things that actually the tools of the cell, they catalyze very important chemical reactions constantly within your body. And really all of this stuff is designed for a healthy cellular function, programs the cell's life, what it's supposed to be doing, synchronizing it with the other 15 trillion cells in your body. And eventually what DNA does too is programs the cell's death and the end of our lives to get ready for the next generation. And hopefully that generation will have the necessary variation to cope with future environments to continue on our species. Next, protein synthesis. This is really what it's all about. Okay, so there's a two-step process. We've looked at it before. There's transcription, translation. Transcription in the nucleus, translation in the ribosome. Together, those two are called protein synthesis, right? And actually, it's under a 3-tier control. There's 3 different levels of DNA that control most of this. We're only going to be dealing with like the lower level, 1 level right now, just to show you the basic principle act of how a message will get transformed into a protein. How that interaction, more complex set of interactions occurs, we'll deal with later, and protein regulation issues at the very last assignment within this lecture series. Okay, so we've been through a lot of this before, but I'm going to add a couple key players in here. Now, one is when you're inside the nucleus and when the DNA is beginning to be translated into RNA, the proteins that do this, we refer to as a transcription initiation complex. There's several key factors going on in there. And I'm not going to hold you to terms like those, but just to recognize there has to be something that catalyzes the copying of DNA. And these are actually enzymes in which DNA produces that feedback that turn the DNA on and regulate its expression, its copying to RNA. The ribosomes, of course, are the things which translate the message in the proteins. Now, the intermediate sort of things that are important for us are the copy of the message. DNA is going to be copied into a molecule known as, again, RNA, and there is a subscript to mRNA. And additionally, when that mRNA gets to the ribosome, something will need to bring the amino acids to the ribosome to be joined together per instruction of the mRNA. And these are small molecules called tRNA, or otherwise transfer RNA. So really that's our laundry list. 4 basic things we need to understand, the processes that surround them to be really successful, in understanding transcription and translation. So a little better schematic here, something simplified, but I think really good. So on the very top, we have DNA. Now, DNA is a double-stranded molecule, and it's tied together by some weak bonds between specific chemicals. So we can look at what the pairing rules are, which are important to establish. There are only 4 chemical nucleotides in there, right? We call bases, but the particular names are going to go adenosine, cytosine, guanine, and thymine, but we don't worry about that. We're shortening it out to just letters. So what are the pairing rules? So A pairs with T, C's pair with G. Doesn't matter which way they go. That's the only combinations which are allowed. No other combinations are allowed within DNA. So that's really how it's zipped together. We'll always see that. Okay? So the first step is DNA is unwound. We have that transcription initiation complex. One component of it unwinds the DNA. Another component separates the strands. And now we have the coding strand, which we're interested in. Then what happens next is the coding strand, letter per letter, is copied into RNA, but we have to remember the copy will be the pairing, okay, the opposite. So look at the middle diagram. So when you see the first letter over here on the left, T, see that a T is not a T. A T doesn't get copied as a T. It gets copied with its corresponding pairing opposite, which is an A. Refer to the very top diagram. You see the pairing opposite is an A. So that is what actually gets copied. It's really the mirror image, right? The corresponding complement. So the next letter word gets a little bit tricky. Now, for all the other letters, C's pair with G's, T's pair with A's, C's pair with G's. We do make a substitution in RNA. With RNA, when we're making an RNA molecule, instead of a T, like we would see in DNA, when we look at DNA up here at the top, you can see where an A pairs with a T. The only difference is when we're creating the RNA complement, our message, is that the A will pair with the U, so we use a U, and that's a substitute. And the reason that this holds true is because think about the name of the molecule. So we have DNA and RNA. Sound very similar. They are. Okay? So ribonucleic acid, that's RNA, and DNA is deoxyribonucleic acid, which means there's just one tiny little oxygen group, one little small chemical change, which makes the pairing rules a little different. The chemical environment is a tiny bit different. So thus we have to have, U as a substitution, and U is actually a chemical, a base in which is called a uracil. So you can, if you're a chemical person, you can add that to your chemical repertoire, uracil. Another reason we want to use RNA. Now, if we think about this too, I should backtrack a little bit here. We're not copying into DNA. See, we actually could. You could actually copy a DNA molecule. There is hardware that would do that. The problem is that DNA likes to be double-stranded. So in other words, as soon as these transcription, initiation, complex members here let go of the DNA, those things snap back together. The chemical bonds snap back together. And this is what DNA likes to be. It likes to be double-stranded. So if we made a copy of this, out of DNA, it would try to travel to the ribosome for expression, so the ribosome could read it, but it would glom on to other single-stranded DNA. It would chemically bind together and be a woolly mess. So the ribosomes themselves wouldn't be able to read the code correctly. Now, RNA, on the other hand, it likes to be single-stranded. It's not going to stick to other molecules of RNA or DNA. So when we make a transcript, we do a transcription of it, it's a substitute for the DNA, it's safe. It's relatively inert, so it can safely get to the RNA, with high fidelity. So next, when we're moving from transcription to translation, moving down, now we're moving out of the world of the nucleus, into the endoplasmic reticulum, heading right to the ribosome. When the RNA gets to the ribosome begins to feed it through 3 letters at a time, because 3 letters as a codon represents 1 amino acid, and the ribosome will sequence amino acids in sequence. Bam! Every 3 that moves through, it'll add a different amino acid on the correct amino acid. Now we want to see how it does. It's a little tricky here how it's actually adding those amino acids on as it's pulling it through. Okay? So let's have a look at how that process works. Now, one thing I will notice here, though, is that the polypeptide strand in which they're looking at here, it is not a mistake. So it's methane, or sorry, it's not methane, it's methionine, it's isoleucine, and serine. So those are 3 amino acids. And that's what that RNA actually codes for. And that's what the DNA codes for. You always have to have a start sequence. There always have to be something to say, to the DNA, where does the beginning of this protein begin? And by the way, a beginning of a protein, is going to be expressed, and the entire length is called a gene. A gene is one functional protein that does something or is a structural component of the body. That's a gene. Okay. There has to be something to say where you start at. Well, in DNA, that's TAC, and that represents methionine. So we see methionine starter right here. And of course, there's going to be a termination codes we have to have too. So at the end of this chain, or the end of the DNA, or the end of the RNA, there must be at least 3 letters together as a codon that represent a chemical agent that says stop the gene here. Okay. So transcription. This is a little bit more complicated, the diagram. It's still very simplified, but it brings us into a little closer, to the action of what's going on here. It reveals some of the transcription factors here too. So I can just mention a couple things. Well, the DNA is unwound by this large enzyme called helicase. And then finally, it's copied by this enzyme called an RNA polymerase. For all intents and purposes, these are like secretaries, doing dictation, in the upper penthouse of a CEO of a corporation. That's what they're doing. They're getting the information, out of the big boss. In this case, they're copying information from DNA, into a piece of RNA, which we're going to subscript with an M and call it messenger RNA. Okay? So you can see how it's doing it one at a time, adding nucleotides one at a time. And then finally, that will be taken out into the endoplasmic reticulum, find the ribosomes. The transcription factors will release the DNA. It'll fold back up. And once it's back together like a slinky again and folded up, it cannot be expressed again. The trick to expressing DNA is all you have to do is unwind it. Once it's unwound, it will automatically express. And once you fold it down, it's shut down and almost absolutely invisible into the cell until you do this to it. All right. So a little different sort of diagram here. This sort of indicates where things are happening at. So we have that mRNA being produced from the DNA as it's unwound. We have the transcription factors copying it off one strand. It's essentially producing the template for it. That mRNA is taken into the cytoplasm, and that's just where all the Estes stuff is. And the endoplasmic reticulum is, of course, one structure in the cytoplasm. And it's taken specifically to the ribosome, the workers. It's fed through the ribosome, and every 3 letters at a time, a corresponding molecule, right? One that has an exact fit of another type of RNA, a very tiny little little piece of RNA that just has 3 letters on one side, will fit like lock and key on top of the mRNA. In fact, it's going to have some opposite pairing rules too, which we already know. So we know that C pairs with G. So let's look at our little thing where it says translation 1, and you see the transfer RNA fitting down. We see that C pairs with G. So there's our lock and key fit. We also see that G pairs with C with the middle rungs, right? So we have a lock and key fit. And in RNA, remember the rules are that A's pair with U. So therefore we have a lock and key fit. So that transfer RNA with GCU as its code, it's carrying a very specific amino acid. And for that one, I believe it's serine. So there will be a different 3-letter code for a different amino acid, all very specific. Thus, next to that tRNA sitting down, there's an empty space. You can see it in the mRNA, and this in the translation 1 diagram. You see UAA. What will happen is another transfer RNA will lock onto it too, and it will have an AUU. You see the opposites, AUU will set down, and it will be carrying a very specific amino acid, and on and on and on. So as that mRNA now set down with amino acids that are being held in place by tRNA is drugged through the ribosome's job, as we can see in translation part 3 at the very bottom, is to chemically attach the amino acids together into a long flowing chain. There would be a technical term for that. We call it a polypeptide chain, which really is just a chain of amino acids that folds up into our functional final product, our final protein that goes to have biological function in the cell. So literally what happens is that you're looking at how chemicals are really turned into flesh and blood. This is our molecular flesh and blood plant. It follows all the wonderful rules of physics and chemistry in the world. It demonstrating that, of course, we are, as organic beings, very much part of the rest of the universe and very much part of the world. I love this. It's a very humbling experience. And I can also see from this that if you were actually, trying to learn how a Tesla works, Teslas like have 30,000 different parts. This is so much simpler than the mechanical world in which we build together, right? I love doing this job because I don't have to think like an engineer and hold, millions of parts in my head. Because once I know this, this is not just for human beings. This is for every form of life on Earth right now, and every form of life practically that has existed from day one is built on this entire model. No matter what organism you open up, its cellular functions in the transcription and translation work exactly the same with the same components. There's variation, of course, in those components. That's what makes all the species and the world different around us. But essentially, it's the same ideas. Okay. Now, a lot of you are thinking you're maybe fuzzy with the tRNA thing. And of course you are. We're adding another layer of complications. We put one mRNA molecule in there as an intermediate, right? That's our first copy and the act of transcription. And in the act of translation, where the mRNAs is being fed through our ribosome, you can see here a closer idea of how these tRNAs are sort of have an anticodon. And it's really what we call those things is the anticodon fit. They'll sit right where it's supposed to, one after the other after the other. And they are chemically stapled together. And you can see why they call these types of small RNAs tRNAs or transfer RNAs. Alas, this is a shot of ribosomes at work. Of course, this is an in vitro shot. We've taken it out of the cell and we've stained them so you can see them. So the green surface here, and you see little dots in there, that's the molecular level. Those are molecules, actually atoms in there. This is a magnificent picture, a scanning electron micrograph. And the endoplasmic reticulum is stained green. That's the factory floor. And the bluish to purplish stuff there, those are the ribosomes. And I love looking at this because it tells us something about the architecture of the factory floor. It's not just like factories. When you go to factories, it's not willy-nilly. You went to a car factory, everything's set up for what needs to happen. Conservation of motion, right? The number of parts per car have to be equal. Now, here's what I mean. So if you're going to make one car, you may need to have one chassis, right? One chassis. How many windows? Okay. There's a 4-door, 2, 4. You're going to need to have 6 windows. For every one chassis, 6 windows. Well, that's the same way it is when you're making parts for the body. So sometimes you'll have a messenger RNA that comes out that won't be just sent to one ribosome. It'll sent to a cluster of ribosomes. And those ribosomes will share the mRNA message, amplifying the proteins to get the number of parts relative to other parts. Now, for example, let's look at the purple ribosomes at the bottom. You see that little filament running and connecting together? That's a piece of RNA. It's being fed through one ribosome to a next ribosome, to a next ribosome, to a next ribosome, producing a number of things, a number of enzymes the body needs. And you can see a cluster of ribosomes right above it strung together. That's where they're feeding, a single mRNA to amplify the signal. But what if DNA doesn't need that much of an enzyme or product made? Where does it send it to? A single ribosome. Look over here. See little single ribosomes on their own? So a piece of mRNA with a different sort of code will go just a single ribosome to make just one thing that the cell needs to keep the ratio of products the same. So it's the organization of this factory floor, like any modern factory, that means efficiency. You don't want to make, 7,000 windows for every car chassis you put. You want to make 6 windows. So this keeps the cell organized. So even at the organizational level of the endoplasmic reticulum, it become clear and apparent that this architecture is set up just right to get the ratios right. Now, we will see variation in this between species. That's what is different. So, we share 99% or so of all of our DNA with our closest primate cousin, which is the chimpanzee. And I'll tell you, people say, what a hell of a difference 1% makes. Absolutely true. But most of that 1% difference in the DNA, it's not so much in the DNA itself, it's how that DNA codes minor differences in the organization of places like the factory floor here. How ribosomes are juxtaposed differently towards each other, where the DNA is amplified differently at different spots or not amplified. So the ratios of products change just slightly. And that in turn will actually produce a lot of variation in the forms. Of course, there are other things we'll look at that make you different from a chimpanzee, but it's just subtle differences in the 1% that can actually do this too. And we'll discover some of that stuff a little bit later in the class. So, transcription and translation, as complicated as it is one of these subjects that I don't want to be labor on. In other words, if I go more than 15 or 20 minutes on the subject, it's overdone. You guys need some cool down time. So take this lecture in, let it cool down, let it sink in, then move on into the laboratory material and start reading the laboratory book, which is going to reiterate a lot of this stuff. And it will provide you with some really good images. One thing I really do like about the lab menu you'll be working in are the solid images in which the authors have used. Okay, hope you've enjoyed. I'll talk to you guys later.